Okay, hello everyone. Welcome to our Peaks webinar series. My name is Caspar and I'm one of the account managers here at BSI. I'm excited to introduce today's speaker, Ching Zhang, our software systems manager here at BSI. Ching will be sharing a powerful and innovative workflow that combines DIA and PRM fully integrated in a single seamless solution using Peaks. Whether you're navigating large-scale discovery or zeroing on sensitive validation, this hybrid workflow offers the best of both worlds without compromise. From deep proteome coverage to precise quantification, you'll learn how to harness the full power of hybrid DIA, developed by Professor Jasper Olsen's group and Thermo Fisher Scientific. At the end, we'll wrap up the webinar with a live Q&A, so please hold your questions until the end. Thank you for joining us today, and now I'll pass it on to Ching for the presentation.
Okay, hello everyone. Can you see my screen now? So first, welcome everyone to today's webinar. So today we will introduce the Peaks. We have a workflow we call hybrid workflow for the hybrid DIA data from Thermo Fisher and Professor Olsen's group. So this is the outline. So at first, I will introduce do a short introduction to the BSI and our Peaks product. Then I will introduce the hybrid DIA method, and we will use a data set from the Professor Olsen's paper as one of our demo projects to show the power of the hybrid DIA. And then we will have the live demo. We will show you the software, and finally we will have the Q&A. So first, I just short introduction to the BSI. In the BSI, I think more people know us as the Peaks. So our company name is the BSI. So basically, we have a lot of our unique features. You may know before that we have the de novo, and now is the time for the AI for deep learning. Now we have the de novo, and now we support a lot of the data type. So basically, we will support the DDA, we support the DIA. Now after the Peaks 12, we support the PRM. And also, since now more and more people are not only limited to these three kinds of acquisition method, they have some we call the hybrid method. So that this is why today we have these topics. We have this topic talk about the hybrid DIA. It's basically just the combination of the DIA and the PRM. So for most of the workflow, we can do the database search, do the spatial library search, do the quantification, and also for the DDA data. We have a very interesting search we call spider search. If we are interested into some mutation or non-canonical peptides, we may try this workflow. And now for BSI, we have two parts of our service. The first part we call this our Peaks software. So maybe you are already familiar with our product. We have the Peaks Studio, it's a standalone product for the bottom proteomics. And we have the Peaks Online. Basically, it's designed for the very large cohort study. For example, we have the customer can run 1,000 DIA samples in the Peaks Online. And we have the Peaks AB designed for the antibody sequencing. And also the Peaks Glycan Finder is more specific to the glycoproteomics and the glycosides. And also we have our lab service. Similar to our product, since we have some unique features for our software. So we have our service for the antibody sequencing, and for the immunopeptide, we have our deep immune service, and also we have the service for the glycan, and of course we still have our general proteomic service. So if you are interested in either our software or our service, we have the contact information in the final slides. You can connect our sales team and maybe our support teams. It's the same. Okay, so now let's start from the basic idea. So it's the mass spectral based proteomics. So this is a recent review from the Professor Mann. So in here we notice that usually for the proteomics, we have the different type of acquisitions, the DDA, DIA, or either the target method. So why we need this kind of the method? So we know that Usually, when we in the discovery phase, we use the discovery method. It's either the DDA or DIA. That means I don't know. I don't have the target in my hand. I just want to see, okay, where are the difference? What's the difference in your sample? And then we have the targeted method. So why we call it the targeted? So that means I already have some targets in my list, something like, okay, I know for this disease, there are some biomarkers. I just want to make sure, okay, in this kind of the patient, this kind of disease, can I find this protein in each or in each of the patients to see, to validate my findings. So now is, I think, in my personal opinion, especially after the 2020, so DIA is more and more popular for the larger cohort study. So you can see every year you can find a lot of the papers to use the DIA method to do the larger cohort study. And this is the rental one from Professor Mann's group. Yes, the 30,000 plasma sample in the DIA run. So why? I think the first is the depth. Since in a DIA search, you can find more and more, and you can find more complete data. So that means based on this data set, the finding is meaningful. Since you can find more protein, you can find the protein more robustness, and then also the reproducibility. Since it's a long time, as you can see here, it's around 30,000 injections. So if the system is not reproducibility, so there are no you cannot find any meaningful protein biomarkers. And then why we need the PRM? So usually still from the same review, so we can know that from the DIA we can find some biomarkers. But if we want to use these biomarkers in the clinical research, there are a lot of the standards before we can use this one. So from discovery biomarkers, we need to do some screen, and we need to set the PRM method. For this PRM method, there are a lot of the steps we need to meet. Something like for the accuracy, linearity, and we also need to give the number of the LQ or LD. So for all these parts, why we would like the PRM method? So basically, if you compare with the DIA method, PRM is more sensitive since it's targeted. It's just a small range to get the result. And also with the linearity, since usually for the PRM, we want to do the quantification, maybe not only the relative quantification, but also the absolute quantification. So that's why we need the PRM. And if the sample is enough, so I think everyone will like this workflow. So start from the discovery, we will find some biomarker. We just use the general proteomics method. We find the biomarker. Okay, we find the potential candidates, and then we will do the validation. So we go into the validation step. We use the we develop the PRM method or the MRM method to decrease the number of the targets, and then finally we use the clinical samples or the large cohort to do the validation to get the final result. So this workflow is well prepared, and a lot of people like this one. But I think in the real practice, the only question is that if the sample is not enough for you to do both the DIA or DIA and the PRM, then what happened? So something like before you do the experiment or before you start your project, you got the sample, you find that the sample is not enough for both DIA and PRM, then what you can do? And also, if something like, okay, since I only have this sample, I can only do the DIA, so I just get the DIA data, I get the result. But then someone will ask, okay, I need the replicates to show the reproducibility. And also, I already know that there are some target peptides in my sample. Why your method didn't tell me that? Is something wrong with your method? And also something like, okay, you give me the discovery data, it's the DI data, but I need to the PRM data to do the validation. So this is the most, I think this is the most situation for in practice, something like we don't have enough sample to meet all these requests. Then what we can do. So that's why the reason the Simon Fisher and the Professor Otis group, they launched this method called the hybrid DIA method. Basically, it's the method to integrate the DIA and the PRM. So just like the figure shows here. At first, we will have a MS1 scan, and then since during the method development, we already have the target peptide list. So if the mass spectra find the target MS1, it will do the DD activation. So get the fractional ions for these target peptides, and we'll compare the fractional ion to see if this is real the target peptides. So during this time, since we want to keep the cycle time, so during this time, it's just we will do the normal normal DIA acquisition. So that means for the instrument, once do the DIA in the back end, it's already have the fractional ions for the target peptides. So we'll do the check. So if the check find that okay, this is our target peptides, then it will trigger a PRM scan. So in the PRM, it will trigger PRM to isolate these ions, and then do the PRM to get the fractional ions. After that, it's just still the DIA scan. So this is the basic idea for the hybrid DIA. It's just a combined DIA and the PRM. So and this time someone will ask, maybe most of you can hear about the shoe-count method. It's used basically the same idea, but the different the one unique features for the hybrid DIA method is here. It can adjust the ion injection time for each ion. So that means if at first we found that okay, for the target ions, the ion intensity, the relative intensity is very low. So that means the sample amount is not enough to generate enough signal for us to analyze. And in this time, the instrument will adjust the ion injection time automatically, something like, okay, in this case, if you just only, by default, you only just use around 4 microseconds for these peptides, the signal is not good enough. But the instrument can help to optimize to increase the ion injection time, something like here. You almost use 100 microseconds. You can see here the signal is more higher. So this is one of the unique features for the hybrid DIA. And then how can we know the idea of this data? So we can in the peaks we have our data view that can help you to visualize this result very easily. So in here, this is when finished acquisition. This is the raw data. In here is RT, and here is the MOV. So we just put all the ions or all the scans here. So different color represent different scans. In here, this line is represented for the DIA window, and for this pink one is represented as the PRM scan. So for each dot here, this is one we call the feature, or we can think of it as the one peptides. So we can give you more details in the next slide. So this is a small time region between it's around the 36 to the 37 minutes, and here is our peak. Is your peak. So basically, the first thing we make sure, okay, for here we have a target peptide. Since it's our spiking peptides, it usually is the select labeled. So and then the intensity is very high. So for us, we will easily find this. We call it's a feature. Basically, it's just you find the peak here. Since we have the MOV here, you have the isotope peaks. You know. Okay, for this peak, it's a peptide, or we call it a peptide feature. Okay, then we can check this feature. So we know this is the MOV for this is the RT, and this is the charge. And also, since here we have the PRM scan, so we go to that. For the instrument, at first it checks since our target, this is we spike the heavy peptides. This is the endogenous, or we have also called a targeted peptides. So we find that, okay, for the instrument at first it's find the target peptide, it's find the label the peptides, and then do the MS2, and then find, check the fragment ion, okay, this is the real peptides. Then it track the PRM scan. You can see here in this time range. While you label the peptides, you look here. So it's in parallel, it's triggered the PRM scan here. So for all this range, you get the PRM scan for these peptides. And in our PRM search result, I will tell you, okay, for this time range, this representative of these peptides. So this is the basic idea. Is for the hybrid DRA. You know that we use the heavy labeled peptides as the internal standard to trigger the PRM scan to find what we call the endogenous peptides, or we call it the targeted peptides. And since we have the reference here, so we know the time range, and also since we have the fragment ion here, so we can based on the fragment ion intensity to adjust the ion injection time to get the better result. Okay, so after this introduction, we know the basic idea of the hybrid DRA. Then the next question that how can we analyze this data? So in the PEAKS, we follow this logic. Usually we have 3 steps. The first step, we do the data split. We split the data into the into the DRA and the PRM, and then we process them in our PRM and the DRA workflow. And then we do the normalization based on injection time. So the logic is here. Okay, we got the data here. We just split into 2 parts. This part is the DRA data. Then this part is the PRM data. Since we will show you later, we have the separate workflow to process this data. So basically, during the process, we have a lot of the AI tools or deep learning tools to help you increase the confidence of the result. Something like here, we will predict the spectra. We'll give the software based on sequence, but predict the spectra. Then we'll compare how the similarity between the measured spectra and the predicted spectra. And also here, we will select the representative fragment ions based on some rules, something like we want to find the fragment ion, most common fragment ions across all the sample. And then, since we mentioned before, in the hybrid DRA, the ion injection time is adjustable, it's tuned. So to compare the result more fairly, so we will do the normalization based on the ion injection time to get the final result. And in the final result, we'll give you 2 parts of the result. So for the quantification result, we'll give you part 1 for the DRA quantification, and then we'll give you the result for the PRM quantification. So this is the basic idea. So in the PEAKS software, usually we offer you a lot of the building, we call the building the workflow. So basically, the workflow can be used based on data type. So this is our workflows for the DDA data. And this is the workflow for the DRA data. And this is the workflow for the PRM, for the hybrid, or for the PRM data. So one thing I want to mention here, so PEAKS itself is when the natural software. So we support most of the vendor instrument and the fragment type here. And also now we support 4 different types of the data. We support the DDA, we support the DRA, PRM, and the hybrid DRA data. So for in the PEAKS software, we always have the concept of we call it the feature-based discovery, just like I mentioned before. So what's the feature? So basically, the feature is all the peaks all the peaks for one peptide you find here. So usually for the DD data, we will use the MS1 to represent the feature. For the DIA, since the quantification, we always use the MS2 to do the quantification. So that means for one peptide, if we want to tell you, okay, this peptide is a content match, we will give you the XIC for both the MS1 and the MS2. And also for the DIA data, sometimes we know that for the DIA data, the background, the spectra is more complex since you just get the fragment ions from a window, not from the single MOV. So the background is more complex. So we want to offer you a course to see or to show that help you to know why I would say that okay, this is something like this fragment ion based on belong to this feature. So in here we give you we call the feature views. In fact, now it's a 3D view. So in the next slide, I will show you the video here. So here this is the measurement peak for this peptide. So let me start my video. So in here we'll give you the 3D view. You can here to adjust use the different angle to see okay this peptide. This is on the this is the other peptide background. You can see okay since they have the shape here, so it's good. More importantly, is in MS2 level. You can see here with colors, it's the fragment ions for this peptide, and you have so many background ions here. Maybe from the different other peptides, or it's just a containment. So we give you this tool to show that why we say this is correct, since you see this profiling, all these fragment ions can be aligned together to belong to this peptide. So we say, okay, this is correct. So this is what we call as the feature view or the 3D feature view to help you to get to make sure the result is confident. So then we just introduce some AI driven models to help you to increase our confidence. So first is the spectra. We can do the MS2 spectra prediction based on the sequence. So in the result, you will have two views. The first is we call the mirror plot. So here in the top is the measured spectra, and then in the bottom is the predict spectra. So you can compare the similarity between these two spectra to see, okay, if this sequence is correct. And also we have the button here. You just click here. You can go to the classical views to check for each fragment ions if they are matched. And if something like for the phosphorylation, we want to check if we have the nature loss ions. This is for the spectra. The other predict we offered is the predict RT. So in the result, usually you will see the RT regression line. And also we give you this give you different figures on the different levels. In here is the sample level. We told you okay for this sample, what's the regression line looks like. And also we give you the idea something like is for this sample in the different range, what's the delta RT looks like. Since we know that okay, here is the start of the gradient, so it's more close. And then in here, something since this usually we use in here the gradient we change the gradient to just wash the column or whatever. So the RT is large, but still can find that even in this part, the delta RT is still very small, usually just 1 minute or 2 minutes. And also in each peptide, we will give you the delta RT for each peptide. You will find, okay, this is the column, this is for the real RT for these peptides. And since we have the predict RT, we can calculate the difference between this predict RT and the real RT to see if this match is correct. So this is our RT prediction model. Also, of course, for the TIMS data, we also have the prediction model for the CCS. And then just something like, okay, we have all these ones, and we just something to show you some benchmark result for the DIA. So it's still, this is a paper from the Professor Alton's groups. It's very famous the ArtShow data, the HYE data. So you can see that for the peaks, not only we can report more protein group, but also you can see the density plot here. the ratio is correct. So we know that, okay, our DI workflow is good. And also, since the hybrid DI have contained a PIM workflow, so we also just to give you a basic idea for the PIX PIM workflow. So basically, for the PIX, the PIM workflow, I want to mention that it's just a very engineered workflow. So it's just one click workflow. It's very easy. Just here in here, you go tell the software what's the group information. Since here we have some spiking some peptides as the calibration curve. So we want to do the absolutely quantification and give tell the software what's the group information, and then just give a transition next. Tell the software, okay, what's the target peptide MOZ, RT, and the charge. Then the software will do all these to pick up the fragment ions automatically to get the final result. So that's why we say it. It's a fully automatic workflow. Okay, then now we know the basic idea or basic performance of the PIX DIA and the PIX PIM workflow. Then we just can go to the hybrid DIA workflow. So in the paper we use the data set is the A549, the peptides is the phosphorylated peptides. It's use a different input different amount, and also there are some spiking the standard peptides as the trigger peptides to get the final result. So in here we show that we use the hybrid DI data. So our target here, we have the target peptides. We want to quantify more the endogenous peptides or target phosphorylated peptides in these samples. And also for the discovery part, since we know that, okay, this is the cell line data, this is the phosphorylated data. We want to see that how many phosphorylated peptides we can find in this background cell line. So in the PIX, this is our workflow. So it's always just a step by step. We offer you the analyze wizard. You can just follow this logic. First, we just upload the data and then tell the software, okay, this is the hybrid DI data. This is the Orbitrap instrument. And then we just select this workflow. It's now for the hybrid workflow, only one workflow, just select this workflow. Then set the parameters. The first part is just set the parameters for the DIA search. So for PIX, we have the idea that streamlined DIA analyze. So you can set either use the library search or just use the database search. Or sometimes when we want to do the troubleshooting, you can even also uncheck all these two. I just want to do the PIM search. It's both fine. All these steps are optional. And then you just set the LFQ, the LFQ settings for the DIA. So this is basically just tell the software for this sample which the group information. And then here then just set the PIM workflow. So compared with the DIA, the same the LFQ group is the same. Tell the software what's the group information. And all the difference is here. Just input a transition next. Tell the software the target peptides MOZ, RT. Then it will do the extraction, signal extraction automatically. Finally, here just is the FDR control. So please note here. So for this result, just for the DI search, since for the PIM now we don't control the FDR, so this is only for the DI result. Then you can just submit the analyze. Then you can get the final result. So in the result node, we call this the result node. So for one hybrid DI search result, usually will give you 3 result nodes. The first one is the DIDB search result, and then is the DIFQ result, and then is the PIM result. So each result node will have the different tabs. Usually we have the first page is the summary page. To tell you the information for the summary information, and then we give you the peptides and the protein informations. So for this part result, since we have the target peptides, we usually want to check the target peptides first. So for this demo project, I just use part of the data set, use 3.2, 3 of the lowest sample input is 2.5 and 10 micrograms. So we can find basically we can find 170 the target peptides. So if you compare the paper, we also have a full data set, compare all these sample input. So you can find 2 more peptides. And also the same as the paper used something like the quality score as the filter. So if we use this score, we call MS2 correlation as the quality filter, we can finally get the 64 peptides. Is close to the result reported in the paper. And then we just want to check the peptides one by one to see, okay, why I say this peptide is correct, and also if the identification is correct, either the quantification is also correct. So for each peptide in the result wheel, we will offer you 5 parts of the result. We will introduce just one by one. So the first part is the peptide information. We give you the basic information and the quantification information here. And for the third, the second and the third part is related to the spectra. We give you the spectra. We have the mirror plot here. We give you the unmatched information here. And the fourth part is the quantification information. We give you the quantity of each peptide in each sample. And then the first part is the XIC part. We give you the XIC for the ferrous ions and also for the MS1. So we just give you more details in the later. Okay. So in the first part is the peptide part. Besides the common sequence, correlation, length, MOD charge. Here we give you we can give you 2 profiling here. We call it sample profiling and group profiling. In the sample profiling, if you mouse over here, you will see the quantity for each of the samples. And for the group, you can see the ratio of the groups. So since we know that this is sample input, so we basically know that okay, this ratio is very close to our expected ratios. And the second part is for the spectra. So we have the mirror plot. We found that this is the real spectra. This is the predicted spectra. In here, in fact, we'll give you two tabs. The first tab is the ion match. In here, we tell you what's the match from the ions and their relative intensity, and this is the we call it query MOZ, and this is the query intensity. And also, you can shift from the ion match to the survey scan here. In the survey scan, we just give you the MS1 spectra. So for this example, you can see here this is the labeled peptides, and this is here it's very small. So I just zoom in here. This is the target peptides. You can see okay. They are also find their MS1 their actual peaks is similar. So this is for the second part. For the third part is the quantity. So for each peptide, we'll give you 2 quantity. So the first one here is the raw area. Yes, just to calculate from the raw XIC, get the raw area. And also, since we know that hybrid DI it have the different ion injection time, so this is the normalized area. So all the ratio are calculated based on this area. And in the final part is the XIC plot. So we offer you the XIC you can click on each samples to see the XIC for each sample. So we offer you the MS2 level and also the MS1 level. And also since we have the multiple samples, and here every time we can see only 1 samples. So we also want to see that to compare across the samples. So here we have the button here. You can click this button to pick up at most. Now we offer you at most 4 samples to do a comparison. And also our y-axis here is adjustable. So I just you can see here for the MS1 it's clear we have the ratio of 1 to this is close to 4. And also, since this is the spiking peptides, so you can see the ratio is close. And since for the x-axis we use the raw area, so I just adjust the y-axis here to give you this basic idea. And also then the next question is that, okay, this is the PRM scans, and how about the performance of these peptides in the DIA search? Since in this data set, the author give us three type of the data. Besides the hybrid DIA, we also have the corresponding the DIA and Shewcomb data. So I just search this peptide, the same peptides in the DIA search result. So you can see here, it's basically you can still find some of the peptides, but obviously you can see here, if in a discovery way. So for these peptides, since the ratio is very close, if you in the lowest amount, sometimes it's hard to find all the sample in all replicates. This is one thing. And also for the ratio, if you compare this ratio to the PRM result, you can find that the PRM result offer you the more correct ratios. So then just the same idea, since for DI, we can check the background ions in the 3D view. So you can see here why sometimes for DI it's hard to find these small peptides, or if the sample amount is very low. Since compare with the background or the interference ions here, our target signal is very low. So that's why you can find that for the same 2.5 microgram the input, PRM can find the result in all of 4 replicates. For the DI, it only can find 2. So that's why something like for this targeted peptides, the PRM use the PRM aggregation method is more can perform well. Okay, we just finished the PRM part. Then we want to talk about the DI part. Since for the targeted, only if we have these targeted peptides, we can trigger the PRM scan to get the result. So, but if you don't have the target list, we just want to use the normal DIA search. And for the DIA, we just want to make sure in the hybrid DIA, we call the DIA part in the hybrid DIA, that this part can get enough, can still find discover enough peptides or enough result compared with if you just use the DIA result. So here we just compare the DI result, DI part in hybrid DI to the corresponding the DI result. So you can see that basically the number are close. And also I do some compare the sequence. Most of them are similar. So that means even in the hybrid DI, the DI part can still cover the same similar dips in with the DI part in the with the conventional DI acquisition. So this is the basic idea. This is the primary result for this demo project. And also for the paper, we have another data set. It's the Cyphos result. It's close to the something like the single cell data. So for the time issue, we will not show the details of projects. We just show one of the example from the paper to show that it have the 10 patterns. So after in the different time points, the ratio of this phosphoryl site is changed. So we can find the same trends in the hybrid DI result if we use the PixPRM workflow. And also this is the same. We just extract the XIC from our result to show the same patterns. And also besides the paper from the Professor Alton's groups, there are still another paper from Professor Bernard's group. So if you are interested, you can connect ourselves. We can talk with you more about this demo project. Okay, now we just go to the software to show you some of the software. Okay, so in the software, this is our result node. If you want to see the progress, so usually at first we will just upload as I show in the screenshot. At first we just upload all the data and select the hybrid DIA workflow, and then we just select okay, this is the workflow. Then we have some basic options. We have the special. You can do either the special library search or the DB search. Let me uncheck this one. Then you just go to the DB search, set the DB search result, and you can okay. Anyway, so we just can also skip this one, and if we want to do FQ, just give the FQ group, and then we go to the PIM. We just here we can input the transition list. So for the transition list, basically it's just we can give you an example. So basically it's just you tell the peptide sequence is optional. Just tell the MOZ, charge, and RT and the window. Then the software can do the extraction automatically, and then go to the result node. Then can submit. You can click on the finish to submit the result. So for the result, usually we have 3 nodes: the DB, LFQ, and the PIM. So for this is the result for the PIM part. We have the summary page to give you the basic idea. You have the heat map here. You also have the volcano plot here and the box plot here to see the result. And finally, you will have the statistical information here to get the basic idea on the project level. And then we have this peptide view to get the result. You can compare the mirror plot, the image tables, and also this XIC. And then we'll have the result from our DIA result. You can just similar to the PIM. You can click on each peptide to see the result, to see the XIC and the area from all for each peptide. Okay, this is the basic. This is the demo for the hybrid DIA. So we just go back. And also more about the DIA workflow. So we also have some demo project, not the HYE, but just the yeast sample in the background, which have some spiking peptides. So we also have the demo project to show that if we are interested, we can also connect ourselves. We can give you some introduction for this the DIA search. And also, I think some user will ask, since we have the hybrid, we can have the DIA and the PIM. Can we also have the DDA and the PIM? In fact, this is the same idea as the show count. We can also in this data set, we also have the show count result. So in the PICS, we can just treat the show count as the PIM search result, and also do the same thing. You can find that for the target parts, the show count can get a similar result, but just to get the less target peptides than the hybrid DIA. So here and just the summary. So basically, the hybrid DIA method is a method that combines the DIA and the PIM. So why we want to combine this one? So that we just want to help the user to get both the discovery result and the targeted result in only one sample injection. Since sometimes the sample is not enough, we can only support it to do one injection. And this method, since it's used the heavy level of peptides as the trigger. So and also it adjusts the injection time. So this will increase the confidence or increase the accuracy and the sensitivity of this method. So now for the PICS Studio after PICS 12, we already fully support the hybrid DIA data directly. By now it's just on the data on the Explorer 480. So we can support you just load the raw data to finish all the process. And also now for PICS we support we have the waterlight workflow to support either the DDA, DIA, PRM and the hybrid. So now it's the hybrid DI data. And for the PICS workflow, we just spread the hybrid DI into the DI and the PRM part, and then process data to get the final result. Finally, this is the reference papers. If you are interested, you can find these papers talk about the hybrid DI. And also we have the poster in for last year's HUPO talk about the hybrid DIA. And then if you are interested, you can connect our sales team to get more information. And also we have a new poster for this year's ASMS poster, also about the hybrid DIA. So we will offer some new upgrades for the results. So if you are interested, just come to our booth in this year's ASMS. Okay, so then this is our contact information. So if you want to learn more, just connect either our support email or our sales email. We can give you more information.